Thanks, Ben, for the kind introduction. It's really a pleasure to be here. I've talked to a bunch of people this morning, really interesting conversations. I mean, I like doing this when I visit a place because, lots and lots of people are using AlphaFold. In fact, most people who work on protein systems probably should be using it or can be using it to figure out how their proteins interact with other molecules. But there are lots of scores that come with it. How do you run it the right way? Which program should you use? All of those sorts of questions come up over and over again. And so in 2024, David Fay at the University of Wyoming in Laramie invited me to come do a workshop. And so I did this sort of 3 or 4 hour workshop there. And in preparation for that, I looked through some of the mathematics behind some of the scores that I think it's like I'm not really sure how this score works. Let's dig up through the papers and figure it out. And some of the scores didn't work the way I thought they did and didn't mean what I thought they meant. And there were ways of thinking about them and modifying them or using them. And so that's part of what I'm going to talk about today. But having these conversations about, okay, here's what your system looks like. Let's do some computations on it and figure out what is going on is something I really enjoy doing. And I have this, that's like my first slide, molecular modeling facility at Fox Chase that we've been running since 2003 where we do structure prediction modeling for anybody at the cancer center. So just like your Huntsman Cancer Center here, Fox Chase is one of the designated cancer centers from NCI. And so we have this core grant funded facility to do structure prediction. And over the years, we've worked on, more than 100 systems, probably a couple of hundred, with 100 publications where, I'm just a co-author in the middle with one of my colleagues, and they published a paper in their journals so that we bring molecular modeling kind of to the masses of biologists in all sorts of different fields. And everything changed in 2021, where we got hold of AlphaFold, where we could do structure predictions that were different from anything that we could do before. So when we might have had 10 projects a year prior to that, now we have about 40 going at once. So the world really did change. Ben's case was particularly interesting because right around when we talked, I ran the current version of AlphaFold Multimer on that system and didn't get models that made any sense to me. And then they retrained it on a later PDB and 2.3 came out. And I ran it and I looked at it and went, that's it. That's the answer. Because you would show me some experimental data on mutations and the helix and et cetera. And oh, the model shows exactly that those are the important amino acids and this is why. And then, the paper just came out in Nature Structural Molecular Biology. It's a really satisfying project where AlphaFold gave us an answer, potential answer that was then validated that really explained a lot of experimental data and helped then plan new experiments to, validate it even further. So, okay. Please feel free to interrupt me if something isn't really clear. We will have time for discussion, but if you're really lost and I've said something that doesn't make sense, please feel free to stop me. So we'll have about an hour now, well, maybe 50 minutes to do the first part of this. And then a 15-minute break, and then another hour, and then a 15-minute break, and another hour. Long afternoon. The last hour probably is when I'll do most of the demonstrating how to use the different web servers for AlphaFold, how to get the scores calculated on some of those web servers, and a little bit of how to use Pymol. If you don't know how to use Pymol or Chimera or any other visualization system, that's something that I think everybody should know how to do if you work on protein systems. I'm going to give you a little introduction. It's not going to be enough. There is a video on my YouTube channel, which has this 2-hour lecture that I recorded right at the beginning of the pandemic. That's all about Pymol. It's just like how to use Pymol for 2 hours. So we don't have that time today. Okay. So when we talk about molecular structure prediction, we're talking about all sorts of different things. So we can be predicting, one thing I learned at a recent conference, just this is totally irrelevant, not related. If you hit Command L on a Mac, you get the laser pointer. And on a Windows machine, Control L, and L for laser pointer. Yeah, I mean, it's down here somewhere in the buttons or whatever. And sometimes I can't find the right button. But the whole Command L thing was a revelation. This AV guy at a conference just said, oh yeah, just hit Command L, and there it is. And Command A is for arrow if you want there. So yeah, anyway, put that aside. So we might do the structure, might predict the structure of a single protein chain. That might tell us something about, what kind of protein we're looking at. We might do protein-protein complexes. These are all structures of aurora kinase bound to a protein called TPX2. It's my favorite crystal structure. Uh, we might want to know how do certain biological ligands or drug molecules might bind to a binding site in a protein, as well as ions here in the middle. We might want to look at post-translational modifications. How do they change the structure of the protein? Maybe later we want to do MV simulations and think about how they change the dynamics of the protein. And of course, proteins bound to other molecules like DNA and RNA. And so these are all the kinds of systems that one might want to do. There are other things to do also, RNA folding, prediction of different conformational states or ensembles of structures, simulations through time. Those are other, computational things one might do. Looking at binding affinities, kinetics, et cetera. There's lots of things that you can do. This is just, I'm not going to go through all of this, but this is, the history that some of which I was part of, over the years of how we do this kind of modeling. So most of what we did prior to AlphaFold was called comparative modeling or homology modeling. You had to find a structure in the protein data bank, crystal structure, NMR structure, whatever it was, that was homologous to your target or targets. Maybe it was a protein-protein complex. You had to align the sequences of your query to that template. And there were lots of different ways, people tested all sorts of different methods for that. You would copy the backbone, you would copy the side chains that were the same in the two proteins. You would model the loops where the loops were different lengths, and you would put on the side chains. Scroll 4 is our program for putting on side chains. And then you might refine the structure. You might do some dynamics of it to see if it might want to relax a little bit. So the earliest example of that is 1969, but in the 80s became more prevalent, and then into the 90s is when it really took off. Another way of doing that was called fold recognition, where if you couldn't find a template by sequence, you could take the sequence of your protein and kind of thread it through the structure of all of the domains in the PDB and add gaps and try to figure out, oh, the hydrophobic residues all end up in the core. You might look at the secondary structure prediction. You might be able to find a template that either was remotely related by evolution or even just the same fold and, it just kind of convergent evolution. That started in the early 90s and was certainly a common way of doing this in the 90s. And then, of course, you have to do the same modeling steps. Fragment assembly came about with Rosetta and iTassor, 1997 and 2007. This is finding like little different pieces and then this kind of ab initio structure prediction, where there wasn't a template in the PDB that you could start with. Could you build up a model out of pieces that might fold into something that looked plausible? It didn't work really well, but it was an attempt. Same thing for protein, doing protein-protein complexes. There was protein-protein docking starting in the 70s. This sort of pipeline, many of these are the common programs that were developed, which is, taking one protein and kind of rolling it all over the surface of the other one. A lot of that was done with fast Fourier transforms as a way to do that quickly. You could do rigid body sampling. You'd have to worry about whether the backbone of the bound proteins was different than the backbone of the monomer proteins if you knew those structures. Protein ligand docking, small molecules into a binding site of a protein. Dock was the earliest of those in 1982. And I was at UCSF and interacted with that group while I was a postdoc, and several other programs. And often that is putting, small spheres into the binding site that might be places that atoms would want to be, hydrophobic interactions or hydrogen bond donors and acceptors, and then taking your list of small molecules and kind of superposing them in there and trying to figure out whether any of them score well. There was never a lot of work actually on protein DNA docking or protein RNA docking that was really specific for those problems. The Haddock and RNP doc were later versions of that were actually specifically trained for that problem. Then AlphaFold happened. So I didn't do it this one, but I have a slide of Galadriel saying, the world has changed. I can feel it in the water. I can feel it in the air. That whole the beginning of one of the Lord of the Rings movies. That's sort of what AlphaFold did to us. Something changed. And the way that came about, I mean, the group at DeepMind that developed AlphaFold in 2020, but the way it was kind of released to the world of biology and to New York Times and newspapers and media was through the CASP competitions. CASP stands for Critical Assessment of Structure Prediction. It was started in 1994 by John Molt, who's a professor at the University of Maryland. He had the foresight to say, well, structure prediction, people are doing structure prediction in the late 80s and early 90s, but we don't know if any of these methods work. And one group will say one thing and somebody else will test their thing and find out that it didn't work very well or it wasn't what it was cut out to be. And so he decided to put together this team, and it's been doing it for the last 32 years. Every 2 years, this meeting happens. And he put this team together to say, okay, so the way the experiment has come to work over the year is, in the winter, the organizers get from crystallographers, cryo-EM people, NMR people, who are determining structures of proteins, protein complexes, and they get the sequences and whatever is in the, it's got DNA in the crystal or whatever it has. He gets that information from voluntarily from experimental groups. Maybe they haven't finished the structure, they haven't released it yet. That's the key thing, it's not released. And so those can become targets that are kind of blind to the predictors. The predictors haven't, the structure's not in the PDB. So in the spring, the targets are released, they're put up on a website. People sign up to be, to compete in this process. And by certain dates, later in the spring, they have to submit their models online. In the early years, just getting the format right of everybody's predictions was a real battle because people would send in real nonsense. There was one group that showed up that had like paper models that they were trying to show the audience of the structure prediction that they did. But that was a group that didn't have real computational resources, but they really had thought about the structure of the protein. Uh, and then every 2 years, they get a different group of assessors who do one for each category of the structure prediction competition. So the comparative modeling category, there was one for the fold recognition category, there was one for the ab initio category where there was no template, different assessors. And those assessors would choose criteria by which, okay, the models are submitted. They now have the crystal structure given to them by the experimental group. How similar or different are the models from the experimental structure? How good are those models? What metrics should we use to say that the models are accurate? And the beauty of this and the wisdom in it was every 2 years, different assessors would choose different things. And so the predictors couldn't learn, well, if I only get this thing right, I'll win the next competition because the next assessor would choose something else. So you couldn't train to just one metric by which to be judged. We were the assessors for CASP 6 and CASP 11. It was a lot of work. It was fun, it was interesting, but a tremendous amount of work. A couple more times John asked me to do it. I'm like, no, it's too much. And then finally, the assessors choose the top, and it's blind. The assessors don't know who the groups are. They're just numbers. And we don't even know the methods that they use. We just have to choose, rank the models, rank the groups on all the targets that they did without knowing anything about who they are and what methods they use, which was also the really good choice because it wasn't like, oh well, I bet Rosetta's going to do well, so oh, this metric Rosetta does better, I'll choose this metric. That's silly and wrong, but a very human thing to do. And so it was a blind test, kind of double blind test. So the assessors would choose the top ranks. We would have a meeting and talk about who should give talks, what the ranking was, and then the meeting is in December, and a year or two later, the papers get published. Long. So this is sort of what happened for ab initio structure prediction, and that means there's no template. So the assessors, the organizers get the structures while the year is going on. So they can get the experimental structure, not released yet, and look at the PDB and figure out, is there anything in the PDB that was related to it? Is the fold already there, or is it just completely new? We've never seen this domain before. So there's this category, sometimes it was called free modeling, template free modeling, and different names over the years. So this graph is about that set of targets at every meeting. And the y-axis is the percent of the model that is correct. I'm going to a little bit later, I'll tell you exactly how this is calculated. It's called GBTTS. But roughly you can think of it as, well, given the target structure and this model, what percentage of the target structure is represented more or less correctly in that model to different resolutions, 1 angstrom, 2 angstrom, 4 angstrom, 8 angstrom. It's a combination of all of those, but it's a percentage of the target structure. So you can see that, when the first meeting happened in 1994, I was a postdoc. I mean, actually, it turned out I was a postdoc at UCSF. The meeting was at Asilomar, which is a conference center in Monterey. And we happened to be there on a retreat the same time the CASP meeting was happening. And we found out about it. I'm like, well, that sounds more interesting than the retreat. So we went over there and kind of watched the first of these meetings more than 30 years ago. But you could see that, you were getting 10 or 15% of it right. That means basically nothing. I mean, a helix or two might coincide with a helix in the structure. But maybe that's just by luck or just by secondary structure prediction. That's almost nothing. There was a long plateau, got a little better. Some fragments you could get right at the sort of 30% level up until about 2014. The blue line is what would happen if the company DeepMind had never developed any of the AlphaFold models. And it was starting to get better that even the other teams that were doing, there wasn't a lot of deep learning, but there were better fragment assemblies. There are probably some minimal deep learning things. They were getting up to 60%. There was progress without the DeepMind group. But the DeepMind group participated in CASP 13 in 2018, did better, but they didn't release code that we could use, so they didn't really have an impact on everybody else. But in 2020, CASP 14 in December, basically they were getting 90% of the structures of all of these brand new folds, completely ab initio, which was just absolutely astonishing. Something many people didn't think would ever happen. And they only vaguely described how they did it at the meeting. But the paper came out in July 2021, and the methods of how they did it made sense and really smart, and people developed lots of programs, related to it based on that performance. If we look at these two proteins, one of them is an ab initio target in that meeting in CASP 14. The other one is it had a homologous template. And I won't quiz the audience as to which one is the one that was ab initio. A lot of people would think, well, it's the simpler one, must be the one that they did from scratch. No, that's not true. The one on the left is the one that they did ab initio, predicted that this, really complicated structure, never been seen before, not in the PDB, with AlphaFold. A structure like that is very convincing. The one on the right is like, well, it's just small, few bit of strands. But the one on the left was really exciting. So how do these programs work? So I'm going to have a few slides on just how any deep learning program works. It's a knowledge-based system. It is learning from experimental data and kind of forecasting or extrapolating, interpolating or extrapolating from that data to new targets. And so it uses all of the they were trained on all the structures in the PDB at the time, but at a certain date cutoff. And there were about 70,000 or so unique, well, 5-6 years ago, 70,000 kind of unique proteins in the PDB. Take all the redundancy out, mutations, very similarly related proteins. There's only about 70,000 that are actually, kind of distinct from each other. So the network takes in the query sequence, the multiple sequence alignment of related proteins. It can also take templates, experimental structures that might be related to the target. So it's kind of a form of homology modeling, and then produces the structure. But here's how deep learning works in general. A deep learning neural network, any kind of neural network, but a deep learning neural network takes some inputs that are numbers usually, or they might be letters. And when they're letters, they have to be encoded. It's called one-hot encode, like a vector. So for the amino acids, every position is either 1 and 19 zeros or 0, 0, 1 and more 0. It's encoded numerically in a sequence. So those are the inputs. And then that's given to a computer and a program that has a function in it that takes those input numbers, multiplies them by some weights, numbers that are trained, does some fancy functions to it, and outputs whatever the prediction might be. So the inputs could be the pixels in a photograph, and the output could be it's a dog or a cat or a tree. That's a classifier. The output can be a number or a set of numbers, just the way coordinates for a structure, that's just a set of numbers. So that's more like a regression, effectively. The function has to have trainable parameters. That's what the W's are. It's trained on real data, so real combinations of x1, x2, x3, and real output y's. So input sequences, output structure coordinates for PDB. And the y obviously can be a vector, just not it's not just one number. So there are tricks that make this whole system work. And every neural network program is basically a combination of these tricks. Maybe there's probably some other things too, but these are the basic ones. The first one is take the inputs and multiply them, just do a linear combination of those inputs. And so I've got 3 inputs. I'm going to calculate a number z1. That is just, weight 1 times x1, weight 1 2 times x2, weight plus weight 1 3 times x3. That's just a linear combination. Same thing for Z2. I'm going to take the same X1, X2, X3, but I'm going to multiply them by 3 different numbers, and I'm going to get a different number output Z2. Mathematically, this is just the multiplication of a vector by a matrix to get another vector. The vectors, let's see. That's one trick. The second trick is, okay, take the output numbers from the after the weights. And the number of output numbers can be larger than the number of input numbers or smaller than the number of input numbers. The dimension of the vector can go up or down. But take those output numbers, and often, you want a numerically stable calculation. You're going to take these numbers and multiply them over and over and over again until it has to be a kind of a complicated system to predict the structure of a protein. You don't want them going off to plus infinity and minus infinity. Be very unstable numerically. And that's what would happen if you didn't do the following, which is you use something called an activation function. And these are 6 of the activation functions that people use that take whatever those input Z1, Z2 are and map them to numbers between -1 and +1 or 0 and +1. So in other words, they scale everything down to the same kind of range of useful numbers. And so the sigmoid function here takes negative numbers and makes them 0, takes positive numbers and makes them something close to 1, and there's a slope in between. And let's see, one that goes to -1. Yeah, this the tanh function here. Negative numbers mostly go to -1, positive numbers mostly go to +1. But there's always a lot of the numbers are in between, and that is useful too. So people choose different activation functions. That's part of the design of the network. Which one's going to work for this problem at this stage of the system? Trick number 3 is to stack these into sets of layers. That's where the deep learning comes from, is you can have many, many layers. And so here we have x1, x2, x3. I multiply through the linear combination with the weights. This is kind of node layer A, and I apply the activation functions. I take those numbers and send them to layer B, where I multiply them by a different set of weights and apply a different or the same kind of activation function. Send it to layer C, do the same thing, send it to layer D, and then finally a linear combination of the outputs of the bottom layer is going to give us the output. You can see that the length of the vectors here at layer A, B, C, and D are not the same. They can go up, they can go down. That's again part of the design of the network to see computer engineers work to figure out what's the best design to give us the solution to the particular problem that we're interested in. I don't know how they do that. I'm not a computer scientist, but they have to design it. And the next step is once you have a particular network you want to try, you have to train it on real inputs X and real outputs Y. And so you really just start by just random numbers for all of the W's. You then calculate for a particular one X point, you calculate, well, what would the Y output be? And usually it'll be nonsense, probably, when you first start training. But then you can figure out by calculating derivatives of the output Y as a function of all of the W's in here and say, well, if I change the W's, this W a little bit up and this W a little bit down and this one really up and this one really down, the output Y will be closer to the actual Y, the experimental Y. And so that's done with standard gradient descent algorithms. And there's always a loss function. It's like, how different is Y output from Y experimental? That's another choice that you have to make. And how well the network behaves and how it behaves is in part determined by whatever the loss function is. The loss being what's the difference between experimental Y and output Y from the network. And so you just do that over and over again. One more trick here is numbers from one layer can be sent back to one of the earlier layers, so you can recycle and go through. That's one of the things AlphaFold does. That's really it. I mean, that's really what all these networks are built of. There are lots of weird and funny combinations and ways of moving stuff around, but they all have to do something kind of close to that. There's one other trick, which isn't really a design of the network, but is more a design of what the network is doing and how it does it and what kind of data it's trained on. And these are called diffusion neural networks. And diffusion neural networks, basically, you're going to generate data information that you're going to train on for the network. And the way the generating of the data is, you take, let's say, a picture of a dog or a cat, and you keep adding noise to it. Or I mean, what I've got here is protein structure. So here's an experimental protein structure. I keep adding random Gaussian noise to all of the atoms, and they move around a little bit, and they still look like the protein. By the time you get down here, they're just a sphere of points. And so what you've done is you've generated a path you've done it in this direction, going right to left. Experimental structure, nice and resolved, to noisy mess. What you're going to give to the network to train it, what you want the network to do, is take a noisy mess and predict a protein structure and sort of denoise and go in the opposite direction. So you're generating the data going right to left, ordered to disordered. You're giving the network, to learn how to do this, the task of denoising random stuff into ordered stuff under certain constraints. You can, as you're doing the training, each one of these. So this has a sequence. This has, multiple sequence alignment, other information about, that also is in the protein structure. So you're denoising it, with the knowledge of what the MSA is. You can then in the forward process give it the MSA and denoise it under that constraint or that extra that information. It's a protein of this length. It has this sequence. It has this multiple sequence alignment. So it's not just, otherwise it might denoise into any, it's like any generative AI. You give it a prompt. You tell it something about what it should do, and it uses everything it has ever seen about things that look like that to start with random stuff and denoise to something that looks like what you asked for. That's it. This is just the network for AlphaFold 2. It takes the input sequence, it gets a whole bunch of related proteins, that's the multiple sequence alignment. This is the MSA representation, which is the length of your protein horizontally, and the number of rows is the number of sequences you have in the sequence alignment. What gets passed through the network layer to layer to layer it's called a tensor. It is you have to think of it as like a cubic matrix, or like a box matrix that has 3 dimensions instead of 2. So the length of the sequence this way, the number of sequences in the multiple sequence alignment, and every one of those, every square in that rectangle is a vector of length 384 or 128. It's just a set of numbers. And those are the sorts of dimensions that are used in this stuff. So that's the multiple sequence representation. The other one is called the pair representation. It's the length of the sequence this way, the length of the sequence this way. And what is going to be in there is going to be something about every amino acid to every other amino acid is going to be something about the relationship of those 2 amino acids to each other, about their distances, about their orientations. But again, all encoded in a set of 128 numbers or 384 numbers. If you looked at those numbers, you wouldn't be able to make sense of them. But the network has been trained to use those to basically to predict the structure. So there's 48 of these blocks that do the same thing over and over and over again, different sets of weights. There's a structure module that finally produces the structure from these representations, and you get the predicted structure as well as all the confidence metrics. And again, you can do this recycling where you get information from this layer and send it back to one of the earlier layers, and that turns out to be very useful. This is sort of how the recycling works. Yeah.
Can you go back for a second? So when that's 48 blocks and that's 8 blocks, is that something that's sort of essential to this process, or was that something that was empirically figured out? Like if we do this 48 times, that it's going to improve the accuracy?
Empirically, yeah. They probably just did different sizes and got the longer you make it, the slower it is. And I don't know how much testing they did, but 48 seemed to be a reasonable number, the time taken versus the accuracy. This is supposed to be a movie, but okay. So this is the recycles as they are going. So the left side is an N-to-C terminal. The right side is the PLDT, which is this accuracy number assigned to every amino acid. We'll talk about it later. But you can see as the recycles go on that the structure kind of converges to its final answer. So the recycling is really necessary.
I have a question.
Yep.
So what is changing if you try with screen recycle? Like
these tensors, these big blocks of numbers, come out of several layers, and then that same block of numbers is sent back to one of the earlier layers, and the process is repeated. In the part of the network that does the recycling, well, if I show so the recycling here, it's just these 2 different representations of you haven't even built the coordinates yet. You're not even thinking about the structure yet. You just have this information in the multiple sequence alignment representation and the pair representation. And those things are recycled to some of the earlier steps. The structure module kind of occurs after that. So basically it's just modification. It's like you're applying a transformation that is hopefully getting you closer to the right answer. And it's almost like you're kind of homing in on the right answer, converging on what might be the most information. And it just turns out that a structure of a network, instead of adding more layers that do the same thing, but with different weights, different activation functions, it turns out to be useful in a lot of programs like this to send the information from some layer all the way back to not necessarily the beginning, but to an earlier layer, and the numbers kind of converge on something. That's the best I can explain it. I think in the structure module, there's probably also a convergent, a recycling step that happens in the structure modules too, where you get a predicted structure, you send it to one of the earlier layers, and it modifies the structure further. I think that's true. Okay. Okay, now ours. There are lots of tricks you can do to make the network behave differently. There's some of this is true during training. Some of it's used when you're actually doing inference, when outside of training, where you take the network and you've got all these weights, all these W's, and you just randomly set some of them to 0, and then you retrain. And that it allows the network training to get over kind of local minimum. If you think of a landscape, sort of a valley here and a deeper valley here and a shallow valley here, and the global minimum is over here, any kind of search pattern, search algorithm might not find that lowest valley, but that's really what you want to find. And so you have to have some way of getting out of the local minimum that are not the global one, and pushing them up here again so that and then fall in the deepest valley. This is a form of that. You just set everything to 0, not everything, but some of the numbers to 0, retrain all the numbers over again, and that allows you to get out of some of the valleys. So the first program was AlphaFold 2. And that just was trained on single proteins and was predicted the structures of single proteins. People did hack it in a way that they got it to do complexes of 2 proteins. But that was superseded only months later by a program called AlphaFold Multimer. They called it AlphaFold Multimer. It's AlphaFold 2. It is AlphaFold 2. The network is the same, but it's the Multimer version of it where it's trained on complexes of proteins. And trained it, and the output Y is supposed to be a complex of proteins, the experimental structure. You have measures between the proteins as well as within each protein, loss functions to tell you how bad the model was and how you should change the weights, the way that I described. So that early version, I was just saying, remember that I've got the command L? There we go. The early version came out in November 2021. There were problems. They fixed some of them in 2.2, but then in December 2022 was 2.3. And the 2.3 was trained on data from the PDB from September 2021, whereas the earlier versions were April 2018. And there's an awful lot I really wish they would just retrain it based on 2026. I don't know why that would be great if they did. I think the program would do better things. This made a huge difference. So the problem I worked on with Ben, these versions didn't do anything. This one gave a really nice answer. That was true of another project, which is this one. And this is with Xavier Grania, who's one of my colleagues at Temple University, of PP2A, which is a phosphatase, a scaffold protein in green, a substrate binding protein called B55 alpha, that's here in blue. We knew the crystal structure of that. There's a small inhibitor protein that's all disordered, we predicted to be disordered, called FAM122A. So the question was, how did it bind to this complex and how did it inhibit things? With that Multimer version 3, I could do a structure prediction of the B55 alpha and this FAM122A, and I got this sort of L shape bound to this blue protein. It was a while later before we had GPUs big enough to do the whole complex. So this is this complex of 4 proteins. The part of this inhibitor protein is sitting in the substrate binding site of the B55 alpha. That's the substrate determining subunit. And the enzyme active site is here where that yellow sphere that you can barely see is the manganese ion in the active site. And this helix kind of blocks the active site directly. We did this in February of 2023, not long after 2.3 came out. The cryo-EM structure came out in October of 2023, and it's basically identical. There is very little information that is present in this cryo-EM structure, which took a long time to do, compared to this AlphaFold model that we did months and months earlier. So it really, that along with Ben's project convinced me that we could do really exciting things with these programs, and especially the more recent one, and really move biology forward and come up with really new hypotheses in important systems. AlphaFold 2 and AlphaFold Multimer, AFM, are really 5 different models. There are 5, all those W's, those are called the weights. There are 5 different sets of weights. And each model in succession was trained on was kind of started from the previous one and then trained in like a different way or different direction, different sets of targets. And so you don't worry about, I mean, these are all the things that go into how the model is trained and what goes into it. And, so one line is important is whether templates are allowed. Templates are crystal structures of related proteins to your target. You align your sequence to the structure, the PDB structure, and you use all of the distances in that template and you take those distances and you feed them to the network. They become additional X1, X2, X3 input numbers to the network from the templates. You can either do that or not do that. And so the early models, the 5 models that you can, if you run CollabFold, if you run AlphaFold today, are 1.1.1 all the way up to 1.2.3. Those are models 1, 2, 3, 4, 5 in CollabFold. The first 2 models allow templates. That's what this second line is. But 3, 4, and 5 do not. They don't take any template information. And that was true of the original AlphaFold 2. It turned out, I only found out maybe a couple of years ago when we were working on one project, that we kept getting good results with no templates and very shallow sequence alignments, even for models 3, 4, now with templates, but with 3, 4, 5. And without templates and with shallow, we should get terrible models. But we were getting really good ones. And so I had to post somewhere. And Sergey Chinnikov responded really quickly and said, no, the AlphaFold Multimer uses templates for all 5 of the 5 models. So we have this project of modeling kinase activation loops with templates that are active. And now we could use all 5 models instead of just model 1 and 2. So that was an important thing to realize. The templates are used. If you're just, I forget to say it later, if you're modeling a complex, you're modeling a single protein, you think you should use the monomer program, the original AlphaFold 2, and not the AlphaFold Multimer. That's not true. Even if you model a single protein, you should use AlphaFold Multimer 2.3. It is trained on a much bigger part of the PDB. All 5 models use templates. So if the templates are important in your modeling process, you should use AlphaFold Multimer 2.3 for everything that you're going to use AlphaFold 2 for. There is no reason at this point to use the original AlphaFold 2. So just that soapbox, I want to make sure that that message is clear. Right. Then, yeah, sorry.
How about AlphaFold 3?
What?
How about AlphaFold 3?
Going to talk about that right now.
Oh, excuse me, I have a question.
Yeah.
You mentioned about the template.
Yeah, go ahead.
The AlphaFold 2 Multimer spell out the IPTM
Yeah.
score.
Yeah.
Is that related with that IPTM score? You mentioned it.
Not directly. If you use templates, you might get a different score than if you don't use templates. And that difference in score might tell you something about the accuracy of the models. Occasionally, if you use templates, like say you use a template that's in one conformation, and you don't know, maybe it's an active conformation, and you want to model the inactive structure. The template might give you models that look active, but what you were really interested in was the inactive one. And even if you use the whole PDB might be dominated by the active structures. Maybe there are no inactive structures. So you're kind of biasing the network towards the template. It's using information from that template. So sometimes it's better not to use a template to see what AlphaFold will do on its own based on everything it's been trained on.
Yeah,
but without the distance constraint. They're not really constraints. They're just added, they're suggestions.
I just try to understand the meaning of the IPTM score.
We will get to that. We will get to that. It's called template modeling score, but it's not really related to the concept of template in AlphaFold 2. It's related, but yeah, I'll get to it. So AlphaFold 3 came out in May of 2024. The paper was published by Nature, and the server, was made public. Initially, they did not provide code. You couldn't run it on your own computers. I reviewed the paper for Nature, and I insisted to the editors that they should release the code in some form so that people can use it. I don't even need the source code. I just needed an executable that will work on my machine so that I can do 50 or 100 models, and I can benchmark it against large number of systems, all sorts of interesting things, develop new protocols for some, if the code was released. So and others caused a lot of drama on Twitter. I don't use Twitter now for science very much. I use it to make fun of GOP politicians. I apologize for that. And occasionally I respond on science, but I do most of my science on Blue Sky and LinkedIn. But anyway, I said I was reviewer number 3, and I told them they should release the code. And everybody's like, "Yeah, why don't they release the code?" The reason is they were beginning to develop drug design things, and they were going to make money off of that, so they didn't want it. Anyway, 5 days after they released it, Pushmi Kohli is one of the senior people, said, "Oh, we're so happy, everybody's so excited. We're going to release the code in 6 months." And they made their deadline by 3 days in November of 2024, which was also a bad time period. But it was good for AlphaFold. We got access to AlphaFold 3 code where we could run it on our own computers. AlphaFold 3 is a diffusion-based structure prediction. So I already showed you, if you generate data from nice ordered things to disordered things or noise, you can use that as training and get the network to take something noisy and build something ordered out of it. So this simulation is just kind of a simulation of how that would work. You start with a sphere of just random points, and then denoise it under information constraints, the sequences, and the multiple sequence alignment, and you build a model of the structure.
Sorry for bothering you again.
Yeah.
Actually, I try to understand the noise. What does the noise mean? I mean, if there is already noise, the system knows the noise. What is the noise?
Yeah.
That means there is already answer the system already recognized. So what does the noise?
The noise means it starts the first after the input. You input the sequence into the network and the multiple sequence alignment. The initial structure is just a random set of spheres. If the protein is 100 residues and there's 300, 500, 500 atoms in a 300 residue protein, that's 300 spheres that are just in random positions in space, centered around some location. And then the denoising process is, okay, well, if the sequence alignment is this, I suppose I don't exactly know the details of this, each sphere is already assigned to an atom. So one of those spheres is the C gamma atom of leucine 32. So it already knows that it should be not very far away from the atoms in residue 33 or residue 31. So that's information that will be used in the denoising process. The next step of the network will bring atoms that are close in sequence together so that now that they're close in space and that their geometries are correct the way proteins are. And then the amino acid contacts between residue 32 and residue 68, because of the sequence alignment, well, those should be close together. So the next denoising one brings those closer together. So it's denoising under constraints, under information that guides the denoising in a particular direction to a good structure prediction. As opposed to, if you said to an AI, just give me a picture. I might give you a picture of anything, but give me a picture of a dog and we'll give you a picture of a dog. Make the dog a tall dog and you'll get a tall dog. The thing about AlphaFold 3 that was most exciting for us and many people was it could do things that were not just the proteins. So it could do post-translational modifications like phosphorylation. It could do complexes of proteins with RNA and DNA and predict the structure of the RNA and the DNA. It could do cofactors, small, organic the web server only did 20 different biological ligands, ATP, GTP, GDP, heme, things like that. Once we finally got the program to use on our own machines, we could do any ligand, any drug, any molecule we want. But the server only does a set of 20. So, that's a lot more biology. We want to model structures of proteins in their most biological context possible. So on the left-hand side here is P53 bound to DNA. So I gave AlphaFold 3 4 copies. I said, give me 4 copies of P53 DNA binding domain. This is a small molecule drug that stabilizes P53. And this is the model, and I gave it the DNA sequence, and that's the model that it built. This was actually with a program called Boltz-2, which is a program trained in 2025. The network is very similar to the way the AlphaFold 3 network was. AlphaFold 3 didn't release the code. When they did give us code, they gave us an executable, not the actual source code, and not the training code. So the Boltz-2 people used the AlphaFold 3 paper, where they in the supplemental is like 75 pages of very detailed pseudocode and equations and algorithms that AlphaFold 3 depended on. So a number of groups have reproduced that by getting their own training data and writing their own code to make a network, but using the same kinds of algorithms. So even though they didn't release the code, they did write very detailed methodologies so that other people could reproduce it with and come up with new programs and not have license restrictions for non-commercial use or commercial use. There's just a much freer version. So Boltz 2 is from out of MIT. It came out in 2025 or 2025 was Boltz 1, more recently Boltz 2. And the advantage is there's no commercial restrictions. So if you work on a molecule that you might want to commercialize, nobody wants to get sued by Google, which owns DeepMind. So you want to run a program that is not AlphaFold 3, unfortunately. But the structure on the right is a ligand in the PDB in pink. And the predicted structure with Boltz 2 in magenta. And you can see it's a pretty good structure prediction and put it on exactly the right spot of the protein in the right orientation. There are differences, but it's a really good structure prediction of that ligand bound to this protein. Ligands don't always work that way. They're not always as good as that, but we'll get to that. AlphaFold 3 has a problem. Oh, it's not a bug, but it's an unpleasant feature, which is every once in a while. So AlphaFold 2, when you ran it on a protein with lots of disorder, would put the disorder all around here like these big lasso loops. That's not what the disordered proteins actually look like. Disordered proteins actually condense quite a lot, some of them. And obviously lots of, they're a random ensemble of conformation. They don't look like big loops out in space like that. But the advantage was this is very, to the human eye, and once you look at a few of these, very signifying, the sort of semiotics of this, I can see the disordered region. It's that big loop up there. You can immediately spot the folded parts and the disordered parts. The problem with AlphaFold 3 is that sometimes it doesn't make these big disordered things. It makes a whole bunch of helices. It makes this like big helical bundle out of those disordered regions. That's hallucination. That's a network hallucinating something that isn't really there. And there are these scores, I'm going to get to them, called PLDDT, one for every residue, that tell you AlphaFold is confident about the neighborhood around that amino acid or it is not. So on the right-hand side, I've colored by that PLDDT. So all these red helices are all the hallucinated parts. They score really poorly, but they do look really structured. So you have to look at an AlphaFold 3 model and look at it, color it by this PLDDT number. I'll explain later how to do that. And make sure that if you see some big helical bundle, it's not completely hallucinated. It's always worth comparing it to the AlphaFold 2 model, which does not really do that at all. So just want to make sure that this, it seems to do it when you put protein complexes together. So this is human TCAT1, choline protein, and RNA. When the RNA is present, you get this hallucination. When the RNA is not present, you don't as much. One thing about AlphaFold 3, this is a technical detail, but maybe a concept worth mentioning, is the idea of tokens. Tokens is what part of a structure are we going to, like how many objects are there in a protein? Well, normally you would think if there's 200 amino acids, there's 200 things, 200 items to think about in the protein. So in a protein, every amino acid is one token. So in the network, the networks are all these big matrices. Every amino acid is, one line in the amino acid 52 is a set of numbers going this way. 53 is the next row, 54 is the next. Each row is a different amino acid. Same thing for nucleotides. Each nucleotide, DNA, RNA, is a single token. But modified, this is one thing they had to do in AlphaFold 3. It was different from AlphaFold 2. We're going to handle modified nucleotides, modified amino acids. We're going to handle ligands. Those are not the same as each other, can't be represented as just a single thing. You need to represent all the details because the details are all different from one ligand to another. So every atom in a modified nucleotide is a single token. And in this is very technical, the PAE matrices that we're going to look at later, every atom has its own row as opposed to every amino acid having its own row. So I hope that distinction is clear. It's a bit technical, but so AlphaFold 2 came out July 2021. This is this chart. I won't go through all of it. By the way, we'll make these slides available. Does anybody want to look at the slides while I'm talking? I mean, look at them on their own computer, because I can give you that link now if you want it. No, okay. Just in case you want to click something, I don't know which one I want. So you will get the slides in PDF and PowerPoint, so don't worry about taking notes. And that allows me to go through some things quickly that I won't spend time on now. So this table is all of the different programs the deep learning programs for structure prediction that have come out. The dates that they came out, whether they do only single proteins or multimers, or they do multipers and post-translational modifications and ligands and DNA, RNA. I had this column that said code. There was a big no here for a long time. I had to change that to yes. With the asterisk that it's not open source, like most of the other things are open source. They did just release the source, actually, just a couple of weeks ago. The source for AlphaFold 3 came out. Not that I don't think the training source came out, but so you can go in and modify it and get more, use it in different ways by being able to have the source code. And almost all of these are available on servers in various sorts. Some of them through Google Colab, some of them as independent servers. Some of them there are several servers that are available. And these are companies that have the different servers. We'll look at that later. Let me do a little bit of this now. This is almost a good place to break, but what?
Up to you, whatever you want to do right now.
Let me make sure I get into this a little bit just for time's sake, so I don't get anxious. So the whole structure prediction field is very old. I showed you that whole CASP timeline and things prior to that. And the CASP assessors had to develop certain measures to determine who's going to win this competition. They don't like to call it, they call it an experiment, not a competition, but everybody who participated behaved like it was a competition. I was the assessor once, and this guy was chasing me through the halls in this conference center in Italy. You must not have gotten the right structure predictions from us, because I know our models are better than the numbers that you just gave out. And did you do this? And did you do that? I'm like, no, leave him. So the assessors have to choose all of these metrics, and the same metrics are used to validate whether AlphaFold was any good, but are also the AlphaFold network outputs some of these metrics as part of its confidence metrics of whether the structure is correct or not. But all of the metrics that it outputs are related to things that came way earlier in the history of computational structural biology. They were used in the CASP experiments mostly. Most of these are developed purely for CASP and sometimes other things. So we'll just do a couple of those now and then take a break. The first of those is pretty obvious that probably any of you who work with structures may already know this. Something called the root mean square deviation. It's very simple. You take two structures of the same protein. You can do it of different proteins, but let's say they're the same sequence, the same protein. One could be a model, one could be a crystal structure, or they could be two different models, or they could be two different crystal structures. Doesn't matter. Two structures of the same protein. You align them by rotating one and translating it so that it aligns as well as possible, so that C alpha of residue 1, C alpha of residue 1 in the two proteins is close together. 2 to 2, 3 to 3, 42 to 42, 100 to 100. As close together in space as possible if I rotate one of them any which way or translate it, move it around. And so, you can have a metric of make those distances as small as possible, all of the sum of those distances, some kind of metric that says, I've got the best alignment. One of the things that's used for that, and one measure of the similarity of the two structures, is called root mean square deviation. All you do is, you rotate one onto the other. I've got residue 1 to 1, 2 to 2, 3 to 3, and I just measure the distances between them. So here's, I don't know, let's say that's 42 to 42. This is 43 to 43. And so there's a distance between, the C alpha atom in one protein at one location of the sequence and the C alpha atom of the same amino acid in the sequence in the other protein. That's just the distance. We're going to index that by j. J equals 1 to n, from 1 to the length of the protein. We're going to square those distances. We're going to add them up. We're going to take the average. We're just going to divide by the number of amino acids, and then we're going to take the square root. That's the root mean square deviation. I always have to think about, well, how do I write down this equation? Root mean square deviation. So I should take the square deviations, I should calculate the mean, and then I should take the square root. That's how I remember the order of these functions. And that's, used in least squares and lots of other mathematical optimization algorithms, but this is just the RMSD. But it depends on the alignment, right? I could use different alignment programs, or I could align just on, this is a kinase. I can align just on the C-terminal domain, the bottom part here, and allow most of the deviation between the two. Maybe the N-terminal domain moves relative to the C-terminal domain. So if I align just on the C-terminal domain and measure the arm, I can align on the C-terminal domain and just measure the RMSD of the N-terminal domain, just to show that that is fluctuating if I keep if I align the C-terminal domain as best as possible. So it does depend on the alignment and the set of atoms that you choose to align. You don't have to align the entire structures, as I just said. You could align just the C-terminal domain and get an alignment and then measure the distances in that domain and in the other domain, or just the other domain. So there's more to it than just aligning the structures and calculating this RMSD. I think now is a good time to take a break. This is going to get slightly more complicated. So let's come back in about 15 minutes, which would be 2:20.